Next up, we have oral presentation 11, University System of Maryland, Maryland. It's a Mission 13 finalist proposal.
Good morning, everyone. So thank you all for coming out today. We are from the University of Maryland. And I'm Lena Franklin, today with two of my co-principal investigators, Avi and Sam. We're going to talk to you about the effect of microgravity on the DNA damage response. Just before we start, we'd like to reach out and thank our teacher facilitator. How are you?
All right, so the experiment we designed concerns what's known as the DNA damage response, or the DDR. The DDR is a network of cellular pathways that sense, signal, and repair lesions to DNA. These lesions in DNA occur as a result of both external and internal stressors, as summarized in the figure shown. So they're exogenous stressors, such as radiation or infection, but most frequently DNA lesions are caused by endogenous forces through regular replication or metabolic processes. The DDR monitors DNA integrity through the use of surveillance proteins, which can activate cell cycle checkpoints and various repair pathways in response to DNA damage in order to prevent potentially deleterious mutations from occurring. A microgravity environment would certainly induce stressors onto DNA. Previous studies in stimulated microgravity and spaceflight conditions have actually conflicted on the response of DNA damage in microgravity. A paper published in 2011 by Rai et al. showed increases in oxidative DNA damage, while a Moreno-Villanueva paper published in 2017 showed little increase in double-stranded DNA breaks. So our experiment was intended to present additional data and determine the effect of microgravity on the DDR, which would certainly be important for people in space, both astronauts and eventual space travel. So to do this, we decided on the nematode C. elegans as a model for the human response. As a multicellular organism with multiple human DDR orthologs, aka homologous gene structures, the C. elegans could provide data on the human response to microgravity. So furthermore, C. elegans has actually been used as a model organism in previous studies, such as with the Gao group or the Moreno-Villanueva group. So once we had determined our model organism, we had to identify the specific genes to study the DNA damage response. We had to pick genes that are known to be part of the DDR, and thanks to this functional genomic map created by Bolton et al., we were easily able to see the interplay between various DDR proteins in C. elegans and determine which orthologs to pick. So we decided to focus on the genes XO3 and MRT2. So XO3 is an ortholog of the human APEX-REF1 gene, and it's a marker for base excision repair and larval viability, while MRT2 is the RAD1 ortholog, which works to maintain chromosomal integrity and promote apoptosis and cell cycle arrest in response to genotoxic agents. Furthermore, these genes have also been shown to have extensive interactions with other parts of the DDR and activate other genes in this pathway.
From all of this, we arrived at our final question. How does microgravity impact DDR and C. elegans in a short-term spaceflight experiment, comparing the expressions of XO3 and MR2 orthologs of human checkpoint genes in a space and ground sample of C. elegans? In order to do this, we divided our test tube into 3 samples. The first contains C. elegans in a dower form. This is a dormant form that allows C. elegans to survive space travel and remain alive until we can activate them. The second is 6 milliliters of liquid growth medium. And this contains everything that a C. elegans needs to live and mature into its adult form. Finally, we have ethanol to be used as a fixative. At U-14, we release clamp A, mixing the growth medium and the dower nematodes. And the nematodes effectively wake up from their dower stage and begin to grow and mature into adults. At U-5, we release our second clamp, and this mixes the nematodes and the ethanol, effectively preserving the nematodes until they can be returned to Earth.
Our proposed ground experiment would replicate the procedures of the space experiment. Ultraviolet lamps would be used to allow the ground experiment to experience the same amount of radiation as the space experiment. This would allow us to make conclusions on how microgravity affects DNA damage response in C. elegans, and thus we will learn how human checkpoint genes may be affected in microgravity. We decided to look at the RNA of the C. elegans in order to determine if XO3 and MRT2 are being upregulated or downregulated when comparing the space and ground samples. In order to analyze the RNA, it must be purified. First, in order to do this, it must be extracted using chloroform extraction. Then, the RNA will be purified with a Cryogen RNeasy MIDI kit. The next step would depend on the quantity of the recovered nematode sample. If large enough, we would perform a northern blot. The northern blot tells us the quantities of certain strands of RNA being produced. If the sample is not large enough, RNA sequencing would be used instead. Through this comparison, we can discover if DNA damage response is significantly altered by microgravity, thus answering our research question. We want to thank everyone at the University of Maryland and our partnering institutions and SSEP who made this possible, including our mentor, How Are You, our advisor, Dr. Eric Hag, and the director of TERPS in Space at Maryland, Natasha Stepanova.
Thank you.
Okay, so this is not necessarily a question. I know that you're a finalist team, but you're listening to research caliber presentation here, which is like really amazing. And I just want to tell a little bit of a story here, just to give you a sense of the power of this program and the heritage. A number of years ago, Howard County Public Schools in Maryland, which is not too far, it's between Baltimore and Washington, D.C., participated in the SSEP program. And there was a young lady at Burley Manor Middle School in the public school system there that was participating. She submitted a proposal. It did not even make finalist. And we found out the other day that she went to the University of Maryland College Park, and she found out that the University of Maryland College Park was doing SSEP as an undergraduate program. So she was really jazzed, and she became part of a team. And now as an undergraduate, she's got a flight experiment going up on Mission 13 coming up this month. So that's a really remarkable history. But there's more. Natasha, where's Natasha? Natasha, you were at the University of Maryland as an undergraduate, and you proposed an experiment that did not get selected for flight. But I remember you were at the poster paper that year, and I came by and you said, we're going to do it again, and we're going to win this time. And you actually did win the flight experiment. Was that Mission 12? And you graduated, and then you became the community program director for the University of Maryland College Park for SSEP. So she now runs the program at the University of Maryland College Park, and we already got funding for Mission 14, which circles back to you, which means that you can propose for Mission 14, and you have to take her attitude. We're going to win this time. Okay, so I just wanted to tell you that, that's a really remarkable story.
Hello, I'm Larissa from Odessa, Texas. How did you determine the appropriate amount of radiation for your UV lights on your ground experiment?
So there's actually passive dosimeters on the ISS that collect information about the radiation doses received in various locations on the ISS. So we are planning to use data collected from those in order to figure out how much radiation to expose our nematode ground sample to.
Thank you.
Do we have any further questions?
Hello, I'm Dale from Maryland, and I'm a little bit out of my depth asking about this question, so don't make fun of me. But I'm kind of gotten into studying about epigenetics recently, and have you guys factored that into this study at all? Basically, what I'm basing this question off of is I've heard recently that through intermittent fasting, you could actually greatly reduce the DNA damage caused by chemotherapy. So chemotherapy patients who fast 2 days prior to chemo and 1 day after actually have a dramatically increased healing rate and dramatically decreased DNA damage. And I'm just wondering if you guys factored that into it in any aspects.
Yeah, so we weren't necessarily considering epigenetics specifically, but Our main motivation was basically to keep the environment in space and ground as similar as possible. So for both, we'd be using the Dower stalk, and we try and keep it on as similar timeline as possible. So that way, to mediate any variables like when nematodes would be awakened, and just make sure that any changes would be due to the microgravity environment itself.
Awesome.
Right.
Thank you very much.
Thank you.